and then which department you're in, and then whether you're just considering RNA-seq or already have data, because that's usually a big difference.
I believe come from Division of Cardiology.
And speak loudly so that they can all hear you.
Okay. And I do not know anything about the RNA sequencing, but yeah, I just want to learn how to analyze this data.
Okay. Yeah.
My name is Felipe. I'm a postdoc at Stephen Mayo's lab, and so I am interested in transcriptional regulation, and for sure dealing with RNA-seq data, chip-seq data, everything would be the focus of my stay here.
My name is Rajesh, and I'm working as a postdoc in pathology and laboratory medicine. And I have RNA sequencing data. I learned quite a bit by YouTube. Now I
want to just go deeper.
Carrie Reese in the lab of chemistry and human genetics, and already have RNA-seq data to work with.
Okay.
My name is me. I'm from Steve Comstock's lab in CDB. I'm a postdoc and I study cell biology.
Okay.
We're from the same lab,
Human germ cells.
Yes, human germ cells.
I'm Shalom Chan. I'm a postdoc from Dr. Harry's lab, and we do a lot of regulation as well.
Okay. In the back, yes.
So, Yuan from Sing Lab in the School of Dentistry. We study bone, and the lab itself has in the past generated RNA-seq data. We can learn more about it.
Okay.
My name is Enrique Sosa. I'm a postdoc in the Clark's lab as well. I work with human and resistant bacteria cells, so I'll be doing that. And I already have one.
Okay.
I'm Felicia. I'm a graduate student at Cedars-Sinai Medical Center, and I'm looking for the analyze my
I am using from Division's lab, and we're looking out for the RNA-seq data. So,
okay.
I'm Katie. I'm from the last lab. I have RNA-seq data that I'm
and we were primarily looking to human cells, and the lab has in the past generated RNA sequences that I'd like to work with and understand a little better, as well as
I'm Todd. I'm in Tom Von Biscuit's lab. We study gene regulation in heart disease.
Okay.
Hi, everyone. My name is Yi Zhang, from a postdoc from University from London. Now I have some data about RNA-seq data and single-cell data. I wanted to learn how to analyze this data and then get the most important information from it.
Okay.
I'm Matt from Nickel Kyle's lab in MIMG, and we developed muscle progenitor cells from pluripotent stem cells, and so we've been trying to better characterize them using RNA-seq.
Hi, I'm Lydia from biology department, and I have some RNA-seq data already, and I kind of played with it, but I'm not sure I did it correctly with them, so that's fine.
I am Cher, also from bioengineering in Dr. Salve's lab, and yeah, I never worked with RNA.
My name is Chow. I'm a student from Machine Lab. We have a whole bunch of chips data and RNA-seq data.
Okay. Here.
My name is Paula. I am a postdoc here at UCLA, and I have some RNA sequencing data from human samples, so I need to analyze.
Okay.
I'm Miran from Shoot Lab. I'm a bottom feeder, so I want to use this time to prep.
Okay.
I'm Eleanor. I'm a grad student, and I plan to generate RNA-seq. I'm Erin, I'm a postdoc in the Carmichael Lab in the Department of Neurology, and we generated some preliminary RNA-seq data and are in the process of generating more.
I'm Nanny. I'm a grad student from neuroscience, and I'm in my third year working in Dr. Video Cohn's lab, and I'm working on single-cell RNA-seq. Now I have some experience with that, but I also want to iterate systematically.
Okay, sure. Okay. Thank you all. And yeah, we have quite a bit of diversity in terms of what people themes are, and even what people's data is.
And before Nicholas start, just one thing that we want that everyone will sign here on the attendance sheet. So just I wanted to put this flag over the room, okay?
This.
G-A-R-N.
Oh yes, I'm sorry, I was muted. I'm not sure when this came back up. Can they hear me?
Yes.
Okay, great. Okay, so let's pause here. and everyone connects to the clusters and requests an interactive session. And we're doing this because it might take a few minutes, and then otherwise we're just going to have to just pause until everyone gets sessions. Yeah, and today we're not going to need a lot of memory. So just as for, I'm going to go back on like what these commands mean just in a bit. Yeah, is everyone who is like currently waiting for the interactive jobs to start? Is everyone connected to the cluster? Who's not yet connected to the cluster? Everyone's connected. Oh, is there an issue? Carolina, I'm going to try to hop back. And has everyone gotten their interactive sessions yet? Yeah. Oh, they should be in the in this folder. So it's Y258VJDN. Okay, so if everyone's good with the cluster, let's just move forward. And so the first thing I just want to mention is basically why are we doing RNA-seq? And this is pretty something you can all have some ideas about. And so basically the point is that we have the genome, which is DNA, and which is not functional in itself. And then there's a number of interaction between proteins and different factors that just make gene expression possible. And then in the end, so we're starting from genomic DNA, and then a bunch of steps happen from transcription to maturation of RNAs. And then we get, for instance, messenger RNAs in cytoplasm, which are going to yield proteins and then perform some function. And usually we're interested more in the function, but we're using RNA as a proxy, even though RNA do not always correlate with protein expression. It's very hard to work with proteins, and so we're using RNA instead. It works well enough, hopefully. Yes. Oh, that's might be. No, I'm not muted at the moment. I was muted just 5 minutes ago, and okay, yeah, we fixed it already. Yeah, and so basically what we're going to do is RNA-seq is just so we have the transcribed RNA, which is then going to be spliced, and typically we're going to just reverse complement this messenger RNA into DNA, and then sequence fragments of that, and then get a sampling of which RNAs are in the cells. So there aren't like RNA-seq is just part of this wider field called functional genomics. So it's just one way to get high throughput data on molecular phenotypes. So really, RNA is a phenotype. It's just like the genotype is just something that's fixed and that's present the same way in all cells of the body, or more or less. And whereas RNA is really correlating with what each tissue does. So it really is a phenotype at the molecular level. And so yeah, so it's just one of the most accessible ones. So I mentioned that proteomics were much harder to do in terms of material terms. And then of course it's also possible to do epigenetics, so ChIP-seq, for instance. And so there are a number of questions that can be asked with RNA-seq. So studying the genotype phenotype map, so how the two relate, how the genotype, just how the DNA just produces different functions, discovering new genes, just characterizing tissues and which proteins are expressed in each, and then working on just like the regulation, how genes are co-regulated, or how the organism responds to some environmental stimuli, or of course, medical diagnostic. So the one point I want to make is that like RNA-seq is very imprecise in terms of just the phrase itself, because we have so many types of RNA in a cell. So the most abundant RNA is actually ribosomal RNAs. And here I've actually put ribosome-related RNAs, so regrouping RNAs, ribosomal RNAs, transfer RNAs, and then small nucleolar RNAs, which are involved in the maturations of ribosomal RNAs. And so basically, because like ribosomes are just like so many in a cell and they're big. So typically our RNA is going to represent maybe 90% of the RNA content in the cell. So all the protocols are going to have this step where they just somehow get rid of the ribosomal RNA. Otherwise, it's just sequencing. Otherwise, 90% of the sequencing would be spent on ribosomal RNAs, which are not very long and which we know the sequences of, and we know the cell, every cell has ribosomes because otherwise it wouldn't function. So yeah, we're going to see in a second, and there are basically two approaches to do this depletion. And then usually the class of RNAs that's going to be the most studied with RNA-seq is messenger RNAs, so protein coding. It is also possible to study like pre-messenger RNAs. And then there's like another set of other RNA classes, so microRNAs, small interfering RNAs, PWE RNAs, long non-coding RNAs, and then transfer-derived RNAs. I'm not sure if you've heard about them. Usually they have a function in like post-transcriptional regulation. So it kind of goes back to this idea that just having the abundances of the different messenger RNAs do not tell you exactly what the protein protein expression levels are going to be, rather than the transcript expression levels. And so one of the main decisions one has to make when preparing an RNA-seq library is to decide how to treat all these different factors that are coming in. So on the left is like this very rough schematic of what the RNA-seq library preparation protocol is. So you just start with cells, and then these cells, they might be just an entire tissue, or they might be a cell type that has been purified by, like for instance, fluorescence sorting, or they might be single cells, in the case of single cell RNA-seq, which is much more expensive. And then, so the RNA is extracted. So we get this like range of different RNAs. And then that's where we can have different approaches depending on what type of RNA the experimenter is willing to focus on. So maybe the most traditional one is poly-A selection. So only messenger RNAs are going to be poly-A enlightened, like for instance, ribosomal RNAs are not, and then most of the regulatory RNAs are not. So just fishing for these poly-A tails will very strongly enrich the libraries for protein coding RNAs. And then the next option, which is generally what is used for total RNA, is just ribodepletion. So there are a number of kits. So it's like ribo 0 from Illumina, and then there is ribo minus from Thermo Fisher. And then the last one is site selection, especially if for people interested in the smaller sizes of RNAs. And then the next step is to just, from these pool of RNAs that have been purified, just reverse transcribe them into DNA. And at this step, there are really two options. And one is, so RNA is only, I mean, genes in the genome are in one direction, right? So the RNA is going to be in one specific direction as well. So really, RNAs are only in one of the two strands on the genome, right? Because genes have a direction. And some of the reverse complementing techniques preserve the strandedness, so you know which of the two strands the RNA was coming from. So you might have areas in the genome where you have RNAs transcribed in both directions. So if you're doing a strand-specific protocol, then you're going to see that you have things expressed in both directions. If you're not preserving the strand information because you're just creating a double stranded DNA from all of the RNAs without preserving which direction they were in, then the two genes that are in reverse directions in the same area are going to be confounded. And this is kind of what this figure shows in the bottom right. So the top part is a stranded preparation, and the bottom one is non-stranded, unstranded preparation. And the strand is always going to be have more resolution to see some patterns and kind of discriminate between different options. Yes, maybe one thing I can mention is that it's possible to do single cell, and then it's possible to do nuclei only, especially if you're looking at like regulation and promoter activity. And then mostly in almost all cases, the sequencing is done with Illumina, which gives you reads that are about 150 base pairs long for like the newer 100 or 150 for the newer machines. But some people do PacBio or nanopore. In some cases, in nanopore, you don't even need the amplification. You can just directly sequence the RNAs themselves. You don't even need the reverse complementation, reverse transcription. Yes. Okay, is this clear? Any questions so far? Okay, so this is what we're going to work with. So basically, this is just an overview of the RNA-seq analysis in general. So it starts with library preparation and then sequencing. And then from that, you retrieve raw reads from the sequencing core. So that's where your work starts. After the sequencing. Typically, you're going to get reads for each samples with separate samples. And then those reads, you will want to just check the quality and then filter them for just remove the bases. That's something cores usually do not do. And then the next step, once you have clean reads, there are different analysis approaches that are possible. The one we're going to focus on is just aligning to an annotated genome. So we're going to see in details what actually that is. So it's basically the genome sequence with where the genes are, but we're going to see the details. And when you have this and the reads, you know where the introns are, so you can kind of put things back together. And when you have reads overlapping exon intron junctions, then you know you have to continue a bit later. So we're going to align the reads to the genome with splice-aware aligners like STAR. And then from the alignments, we're going to count the counts of every gene. And then next week, we're going to do the analysis of the expression matrix. Just quickly to mention the other options that are possible. So this is just taking the reference genome with where the exons are, right? And then the other option is, one is just to not have a reference genome at all, just to assemble the de novo transcriptome fully. Or sometimes it's possible to de novo assemble transcript only based on an existing genome. And then the last option is just to, from the annotated genome, create a transcript database and then align to the transcript database and then work with that. And typically those two other approaches are going to be used for more precise questions, not for differential gene expression, for instance, but we can talk about it if you have questions. So yeah, so this is what we're going to go over. This is just like a kind of reordered diagram. So from raw reads to per gene count. And so basically the middle stage is going to be, so we start with raw reads, and then the middle stage is going to be having reads aligned to exons and to genes. So we're going to get measures of the expression for each gene, which we're just going to then use, I mean, in this case, the program called HTC count to create the, to just transform our read alignments into a table of counts for every gene, for each sample, right? Is this clear? Does this table make sense for everyone? So we have a number of genes, let's say 20,000, and then we have a number of samples that depend on your experiment. And then you'll see that each gene is expressed at a certain level in every sample, and you're just going to have a single value for that. So this is like gene-wise expression counts. Okay, is this clear? Okay. So yeah, basically that's where we're going, and yeah. So any questions on the RNA-seq overview part? Well, if not, let's switch to just the Hoffman-2 intro, which so I have put in the Google Drive. I've also put. I'm not sure how experienced you are all with shell commands. So there is a shell command reference in the Google Drive, which you may want to open, like which is the basic commands if you need a refreshing, need to refresh your memory, or at any point, just feel free to go back to it as we're working on the cluster. So generally, you've all been on this page when you ask for your account. So this is the main page you should be aware of regarding the cluster. Just that's the web page of the cluster itself. And they've got like a frequently asked questions in case you run into different problems, and it may or may not be in the list, but there's a good chance like basic problems are. Yes, so basically the essential grid commands. So just SSH in, SSH is secure shell. So a shell is just like what you get when you open a terminal. So there's SH, stands for shell. And then their main one is pretty bash. And then SSH is just secure shell. So it's like encrypted, just an encrypted way of communicating with a cluster. And then once you're connected to the cluster, so your terminal is effectively going to be working like on one of the machines on the cluster, meaning that if you like add letters, it's actually going to be processed by that machine over there, and that's why sometimes you'll see some delay when you type letters if just the connection is kind of shaky. You're just going to type things and it's not going to appear. It's just because things need to go back and forth. Like the key presses need to go to the machine over there, be recorded, and then be sent back. So that, yeah, that's what SSH does basically. So once you're in the cluster is basically a grid of computers. So just the network of computers that are all linked together, and they're all going to have the same software. They're all going to share the same hard disks. This, when you just SSH into this Hoffman2.idre.ucla.edu address, it's actually going to send you to a logging node. A node is one computer in the cluster. The same thing as one computer, one machine. And when you SSH in originally, you get into a logging node. And that's not a node that's made to do computations. So the first thing you want to do is what we just did, just ask for an interactive node. That's going to send you, again, like ping you off to another machine that is a compute node and which has like available processing capabilities. And you're just going to reserve a processor, basically. And with some memory you have asked. The other way to do this, so this is to do it, QRSH is to do it interactively, and then there is QSub, which is to send a script to the cluster, and then the cluster is going to record that you have submitted a script, and then it's going to find available time for the script to run, and it's going to send it to a machine, and then once there, the script's going to run, and then the cluster is going to inform you back when the script's done. And yeah, so QRSH is interactive, QSub is for batch jobs. And then here is just like the main, maybe resource options you're going to work with. So these are the two we've used just before. So there, you can use them with the L option, so little L, and then H_RT, and then just a number of hours you want. So here, because interactive jobs, and we're not going to be here in 4 hours, we're all going to have closed our computers. We definitely don't need more than 4. On the standard submission system, you can ask for up to 24 hours. And then the second main option is memory. And then same thing, you just ask into the -L option for H data. And then you ask how much RAM you want. And the last one I want to mention is IP. I'm not sure if some of you, and some of you do, not all of you, you have access to a computing queue that your lab has paid for because your lab had both nodes on the cluster. And then you get relaxed, you get like more computation resources in a way or another. Because the last two commands maybe is my jobs. That's just to see which jobs you currently have running. So if you're into an interactive job, you're going to see that you have at least one job running because that's your interactive job. And then for bad jobs, they might be queuing or they might be running. And then after they complete, you don't see them anymore. And the last one is there are a good number of programs that are on the cluster, but that are not directly accessible and that you have to load with module avail. So for example, Python on the cluster, on the Hoffman 2 cluster, you generally have to load Python before you can use Python. Just a few other things. So yeah, I mentioned the login node. So the first node you arrive on when you SSH in are not for computations. So don't run anything. You can run you can browse files or just submit jobs, but don't start I don't know, running expensive software, computationally expensive software on the login node. So just ask for an interactive session right away. And then disk space. So basically that's where you have storage space on the cluster. And there is your home directory. So which is going to be, this actually /u/home here. Yeah, and that is, that's one place you can use, but it's limited to 20 gigs. So usually you want to use your home directory for install programs or general documents, but data is it's going to be too small for data. What we're going to work with in the workshops is a scratch space where the limit is 2 terabytes. But the scratch space is not a permanent space. It's really it's really considered a temporary space. So after 2 weeks, any file that's been in the scratch space for 2 weeks and not modified or updated is considered forfeit and maybe remove it at any time. So anything you have on the scratch space, you can't consider, you just really have to know might be just removed at any time. So don't leave anything precious in the scratch space that's not also present elsewhere. because it's not reliable. It's also not backed. And so many of you who already have people in your labs who have worked with NGS data, you probably have lab space. So this project space, which has been purchased and is like updated every year. And finally, just so the workshop data, that's where I put it. That's where we have the raw reads for analysis. And so in the Scratch space, in my Scratch space, I think you should be able to access that directory directly. Yeah, and then we're going to jump to that. Okay. So yeah, have you all been given an interactive session on the cluster? Yeah. I mean, sometimes it's just depending on the load, it might take some time. Oh yeah, actually I skipped this. Just transferring files, it's done like SSH is a secure in the secure shell. There is SCP, which is secure copy. CP is the copy command for Unix shells. And then you can use it either command line, which is the simplest way, I would say, or you can just install a number of software that will just do it for you. So especially on Windows, you will probably want to do that. Unless you use, I haven't mentioned it, but on Windows, if you have Windows 10, and you're going to do a lot of work on the cluster, I would recommend installing, like activating, I think you need to activate developer mode, but then you can just go to the Windows store and install something called Windows Subsystem for Linux. And basically, you're going to have a full Linux environment in your Windows, and all these commands are going to be available to you, and you're going to be able to apply all the tutorials you see online for Linuxes or Unixes, Unix and that. Yes, so I'm going to talk, the next thing is to talk about the FastQ format, but let's just go to this, so go to this directory here for where the workshop data is, and then we'll start looking at the files. Can you access the files? So for now, there should just be a list of samples and then all of the raw reads. So you can see the files, it's not giving you permission issues. Okay, so you may want to copy the files to your own Scratch space, which is going to be $scratch. If you use $scratch, like the $scratch variable, that has the path to your own Scratch space, like the $home variable has a path to your own home directory. The $scratch variable, capital Scratch, has a path to your Scratch space. So yeah, just go to your Scratch space, create a directory for the workshop, and then copy the file or symlink them.
You're supposed to type the last line of command.
Yes, CD there, and then you're going to be able to see the files. Who's comfortable with FASTQ files already? So you want me to? Yeah, okay. That was the next thing, so it's good. Yeah. Yes. Okay, so I think we're going to spend a bit of time on this because I think most of you don't have like a huge experience with just moving around with command line. So I do expect it to take maybe a little bit of time, but that's why we're here. Oh, don't use QSoup. So. Oh, you have a space here. There was an error. Okay, with command lines, generally, anytime you see an error, you want to pause, and then there is going to be a reason why there was an error. And then you want to understand the reason for the error before you move forward, and what happened and why what you thought you were doing did not work. So actually, this is, can you do Control C? Try Control V. Control V instead of Control C. Has this worked? Right? Okay, great. Then type the QRSH command again. So the reason why, so you're going to see in the drive folder, I have a separate PDF for the, like, just using Hoffman 2, and that's just because I had this slide to connect this morning, in the beginning of this PowerPoint, but today, tomorrow, and on Thursday we're not going to have that. So just go back anytime you need to the PDF for Hoffman 2 usage. Where is it going to kick you out? Try having these two things, like L, H, R, T, and then H data. But if you have a space, it's going to be confused by that. Do you want to do Control T? Control T. Yes. Okay, so go back. Oh, up arrow is going to, in principle, going to show the previous command. Up arrow. Oh, it will be too. That's where you're currently always where the green thing is. So go up arrow is going to bring the older commands. Do up arrow, just up arrow. Yeah. So this one, and then remove the space and enter it. So H. After the comma, there was a space. So that was a problem. Oh, you need to. It's because it's trying. Is a directory means this is. It basically is telling you this is not a program. It tells you this is a directory, but what it really means is this is not a program. Because the command you want to use is cd, change directory. So do you have the? So go at the beginning of the line, and then cd, and then space. Just to make. If you do this, it's going to change the directory to the directory you're giving to that directory.
Oh.
And why is this not?
Oh yeah. And now you go for the CPU flash scratch.
Like this. Okay, so just to show you what I meant is that, and can the people online see the terminal?
I will double check. I think so because I see it.
Oh, you see it. Okay, great.
Yes, I see it.
so you have this. So PWD just tells you, I mean, really a terminal is just like your Windows Explorer or your Finder. So you're just, at any time, you're just in a, just as if you had a specific directory open. So for now, I'm in this directory here, which is actually my home directory. And then I can do ls, and this is what's in my home directory, just because it's not much. It's just because most of my data is in the project space for my lab. And then I can go, actually, if I can just go to a different directory with, it's like double clicking on the directory in Finder is going to take you to the next directory, or just clicking on a directory you've put in the shortcut. For instance, you just click there, it takes you to that directory. And then usually you have so in Windows, you're going to have this bar at the top that tells you like the path with backslashes, the path of the directory you're in. And in Mac, it's at the bottom of the Finder. It just tells you the different directories that are above the one you're looking at. Oh, one thing, the tab key in terminals, it just auto-completes the words you're typing. So that's a tab key. Anytime so I can just type /u/f, and because flash crash is the only directory under /u, so there's a /u directory. Then under that, I can go to the /u directory if I want. And then ls is just printing the list. Ls is for list files. It's actually list segments, but it doesn't mean anything. It's list files. So it's all the files, and then it appears blue because they're all directories. And then flash crash here is the only one that starts with an F. So if I do cdf tab, it's actually just expanding it. And I also know that flash crash is not going to have a typo in it because it was completed automatically, which is very useful. And then, yeah, so here, if I try to complete with tab after the n in the flash crash /n directory, it's actually not completing because there are 100 directories starting. What's going on there? I have no idea what's going on. What is it? So it's telling me they're not seeing it. How do I bring it to the front? This is the question. So this is still doing the same thing as this. I'm doing this. No, it's not. Could you ask for a lot? Thanks. Okay, so we're trying to just autocomplete this n here. So I'm looking for n Rochette. Well, actually, like there are 100 directories because there are 100 users whose names start with n. So it's just asking me whether. So I can either just say I want everything. Well, actually, I cancel it. Anyway, if I do nr, it just completes it. Okay. And then this is what's in there.
Oh, it's frozen.
Yes, it's frozen.
No, it's telling me something weird. I'm not sure why this is. Oh, this is here. Resume share. I was just able to resume share for some reason.
Yes.
I don't know what's going on.
He forgot to resume share when he goes up, switch over to from one thing to another.
Press share.
Yeah, so in this case, when he has to put that code in right there. And
I think it's working, all right?
Okay.
Yes,
Okay.
so now it's okay, yes.
Okay,
They see it, they wrote.
when you go to a different program, it will show.
Okay, and this is what's in the directory. So this FastQ file, it says FastQ directories, and then this samples.list. Also, one of the very important commands, and then samples.list is in my case, white. It might be, if you have a white background, it's going to be black in your case. So it's a regular file. And it's actually just a text file. One of the most useful commands is less, which is just a text viewer, like your Notepad or Windows Notes. There's the content of the file, and so it's just the sample names. One other command that's very useful is WC, and I think you should have in the command reference in the Google Drive, there should be like a list of commands like this. Not all of them. It's not perfect. It's not necessarily meant for your profile, but that's the one I found that was, I think, the best. Yeah, and so if I do this, the first number is just a number of lines. So in the next case, that's going to be the number of samples. So we're working with 30 samples. And if we go to the FastQ directory, so these are all the files. And so they're .fastq.gz files, and then there's one for each sample. These are the sample names, the first parts of the names. Okay, so try to copy this directory and then this this file. And basically what you can do is, if you're in this directory, you can do copy R. For R means recursive, means this directory and everything that's under it. And then / means this directory. And then something like scratch. Right? So if you just do $scratch, it's going to give your own path. So in my case, it doesn't make sense because it's already where the directory is, because that's just my scratch space. But yeah, basically that's what you want to do. Okay. And then what we're going to do is work with the first of these FastQ files. So this one for now. I think I think I just took the first in the. So yeah, any questions? Does it make sense? Okay, so let's switch back to the. Okay. Okay, so the FastQ format. So it is a plain text format. So it's going to be like a txt file in principle. If you have a .fastq file, and sometimes it's .fq, you're going to be able to open it with Notepad or any of these text viewing softwares. Except they might, very often, I mean, those FastQ, they're going to be gigantic files, several gigabytes, and then the software may complain a bit. Depending on which one, they may complain a bit about, they may just freeze because the file's too big. But in principle, they're able to open it, right? Just to take, what's the name in Windows? Just, yeah, Notepad or Text Wrangler in macOS. And you can just see the FastQ file. And usually they're compressed with this program called gzip, which is the most commonly used compression program in the Unix environment. And basically, this is what the FastQ file is going to look like. And you should be able to see this with on the cluster with this file. If I switch back, what I mean is, I switch back to this. Okay. So I have this, and then so I have this file. I'm just doing copy paste here. One shortcut that's useful in terminals is Control-A and Control-E. Control-A brings you to the beginning of the line, Control-E to the end of the line. So you can just paste your file name. And then I can do a command called zcat. So it's like the cat command, except it's going to work on gzip files. And then I can just view it with, I mean, I can just print the first 10 lines with head. And this is my FastQ file. So yeah, so this is the first line, this is the second line, third line, and fourth line. And yeah, so basically what we're seeing here is what I've highlighted here. So this is FASTQ format. It's just a text format, and then you have one read corresponds that's coded in 4 lines, and then the first line is just the ID line, and then you have the sequence itself of the read, and then there is a spacer line. I'm not sure exactly when it was added or why, and then there's the read qualities in the Fred format. So yeah, so one read, 4 lines, and then you have. So first 4 lines is the first read, and then you have lines 5 to 9, the second read, 5 to 8, I guess, and then etc. for as many reads as the sample has. Just to come back to this Fred scale, so the FastQ scores, so which is the 4th line of each read, right? So we've got this like mix of weird characters, and it's actually the encoding that developed for this FastQ format. And it is based on Fred scores, which are log scale 0 to 40 scores. So 0 being bad quality and 40 being great quality. And basically the way the scores work is that if you're for a given base, so you have as many quality scores as you have bases here. So this a mono font, so all the letters is the same size, so everything stays aligned, and you can see that you have as many qualities as you have bases. So for each base, you have a quality score that's encoded with 1 character. And then this character corresponds to a number. I'm going to just after this. And then if this number is 10, then you just apply this formula. And so score by 10 is 1. And then so it's going to be 10 to the minus 1 is going to be 0.1. So, and that's give you the probability that the call is false, that the call is wrong. And so if you have a Fred score of 10, that means you have 10% chances that the base is wrong. Then a Fred score of 20 means 1% chance that the base is wrong. And then same thing for 30 is 0.1%. And then that means that the probability that the base is correctly called is, so for instance, for 30 is 1 minus that. So for instance, for 30, the probability that it's false is 0.1%. So the probability that's right is 99.9%, right? So basically that's how the Fred scale works. It's just a log scale between 0 and 40 that gives you the probability that the base is either false or good if you do 1 minus. Between 0 and 1. So it's probability of a good call just with this log scale. And then the way the characters are coded is usually using this ASCII code, which is just a way to write just letters and all sorts of characters on 1 byte. So it's 1 byte lets you have numbers from 0 to 255. And basically just because, so all these first numbers are actually non-characters, and actually there's space and there's tab somewhere, but most of them are just things that you don't have to be concerned about, but you can't write them in text files, is the point. Then starting at 33, you have this, you just have punctuation first, and then you have numbers, then you have some sort of punctuation again, and then you have capital letters, right? And so the most common way FastQ files are encoded, and nowadays it's fairly standard, at some point it has varied a bit, but nowadays you're going to find Fred 33, which means that the 0 is a 33. The 0 is going to be the first printable character. So whenever you see an exclamation mark, it means a Fred score of 0, meaning a very bad call. And then on contrary, if you see an I or any of the capital letters, it means that this is 33 and this goes to 73, so that's 40, that's 0 to 40. And then any of the capital letters mean something that's close to 40, so very good quality scores. And then, so this 5 is actually 20, so that's going to be, so 53. So that's 53 minus 23, which is the start of the scale. That's 20. And then question mark is 30. So 30 is, if you see a question mark, it's actually a good call, right? And it's just this based on this ASCII code, which is used like in Windows and in Unix, and which is fairly standard in all, like even across like languages, and it's also present in Unicode and everything. So yeah, that's basically how they made it. They made a number from 0 to 40 fit on a single character. That basically was the point, and why they had to resort to this weird thing that introduced like punctuation and weird characters in the files. So really these, like this force line here, it's just each symbol is a number from 0 to 40 using this ASCII table here. Okay. Okay, so here's here are a couple just short exercises to do with this FastQ files. So yeah, just how many reads are there in the file? And then how long is the first read? And then are the base calls good for the second read? And we're working with this file here, p10ko rep1. And in a second, we're going to go over the source study, so you'll know what this name refers to. But yeah, let's just work with this file for now. Yes. Oh, so less is great, but you have to know the shortcuts a bit. Like to quit the less screen is actually Q. So yeah, you just press Q and it's going to quit the software. And then yeah, there are a few, like D is scrolling down, or you can scroll up. I think D for down scrolls down, and U for up scrolls up in less. And there's a number of other things you can do, like changing like tab widths and printing lines on one line or wrapping them. Yeah, just Q to exit less. Good hand issues. Is great. Are they happy? Oh, it's just because it was frozen. So actually, if you use less, I think, I'm not sure if it's configured like this by default, but less is going to drop in a JSON file with less. It's going to know it's a JSON file and it's going to exit it for you, then exit. So you don't have to use any other, like the two commands you can, there are a number of commands you can use and compress JSON files, and they're really all the JSON commands. There's zcat, which all the JSON commands, the other hood, and then there's JSON dash cd, actually there's the same thing as zcat. Then there's g unzip dash cd, which also does the same thing. Zcat is maybe the just something to remember. Does the same thing as cat, which is for zip file. And cat just prints the files. What you need is control. So the trial is so this is what is it going back to this This is not actually the cluster chart, just the and you can just work as long as you don't modify anything, you surround with your yeah, because you want to make so when you chat, so I don't have it. Unless it's one of these videos that you Oh, actually, the question mark in the flashback directory is just like an unknown character. Question mark means one unknown character dash or shell. And really, that character should be. It is going to work. Actually, is the way if you type the question mark, it is going to work. Just you're going to find the right character that because there is only one way to with your username. It is actually the first letter of your username. Question mark. So if you want to do the transfer in the background, so one way you can do it is first open a different session on the cluster, and you don't have to get an interactive job. You can just copying files. You can just do it on the login nodes. it's fine. Yeah, so you can open a second terminal and then do the copying in this second terminal, and you don't have to wait. The other thing you can do, where's my mouse? The other thing you can do is, for instance, if I want to copy this row of FastQs in the directory above this, so I'm just copying I can't copy the entire directory like you are, because otherwise we'll just move it to the same place. It would tell me the same directory anyway. So let's say I want to copy all the FastQs to the directory above this one. And that's going to take time, right? So I can interrupt it. And one thing I can do instead of doing this, so that I don't have to wait, is add this commercial and sign, and it's going to run it in the background. We have not removed all the files for the workshop. No, I have not. Oh, one also useful, very useful shortcut in Bash is Control-R, which lets you search your command history. So if I do Control-R CP, it's going to bring back my copy command. And so I can add this and sign at the end. It's going to run it in the background. So it's doing the copy, just it's not actually using up my, and it's not blocking my terminal. And so this is so it's copying one file, and I'm not sure why it's waiting there. Yeah, it's copied 2 files and everything. And if I do %, it actually brings it back to the front, and then terminal is reserved for that command again. I can interrupt it. Okay, so if copying takes too much time, just put it in the background with the @ sign at the very end of the command. With the @ sign. Not the @ sign, the ends. Oh, this is because you can't copy to the above directory, the dot directory, so directory above the directory to work on. That's my batch itself, which can have function. So copy to your own batch. So our batch you have your own. So this will repeat the dash on after SCP, but that's each copy over the files. So the first time the so you can, if you want to just no, to your own batch. So you can only see from the on the browser context just because of the way the movie is. So don't use S. Just if you control C, you know those branch. Oh, it codes in H. That's your stuff. As I was saying before, the question was this section. So you want to copy the workshop directory to your branch. So just to back the last few questions. Okay. Oh, command not found means this is not the command. Oh, this is actually part of the sentence. Like what's in the Calibri font is part of the sentence, and then just the name of the file is something that should go in the terminal. I mean, the whole line. No, the whole line is not a command. It's just part of the sentence. It's just. Oh, okay. So use less. So if you do less the file. Oh, less LESS. And then the file name. Tab. Oh, P10K and then tab. And then 1 and then tab. That should work, I think. Yeah. And then Q to quit unless. Yeah. Like Q is the first letter of quit is yes. When you see this sort of things, because you're actually looking at the compressed files, they're also compressed files. Is this head? Oh, tail. Yeah. So tail and head is the same thing, but the problem with tail is that if you do gzip, then before you can show the last typeface, it had to compress into a file. So it can take a long time to do tail. It will work eventually, but it should take a long time because you have to go through the interface. So gzip is actually going to in this case it's going to contain. It's going to tell you you don't have the machine. So it's gzip or use zcat instead of gzip. Yes. And this is zcat is the same as gzip, just so you were able to answer the questions, or 3 questions? Not yet. So maybe the easiest of these 3 questions is a third one. So if you just take the FastQ slides, in principle, you should be able to answer the third question. So this is kind of the third question. I'd like everyone to be able to kind of give it a try, a real try by themselves. that's actually right? Oh, so this is 32. So this way you can copy to scratch code. So that's with the SCP command. And the SSH, you want to run them on your Mac. So you want to have connected the terminal. You want your terminal working on your directory. Position directory is log is on your Mac. And that's when you want to use this. Once you CD to that directory, then SCP is files or directory. Oh, I think there's one command that's not in the Unix, which I'm pretty should have been the very first one. The very first command in the Unix command reference, and that is the manual command. If I do manual for SCP, then it gives this entire description. Description is actually open in less in this case. So I can scroll down with D or scroll up with U. And when I'm done, I just press Q to close it. And then those lines that printed is actually my computer telling me that, or the cluster telling me that the copy of the FastQ files is done, which was running in the background since before, right? So yeah, so this is a manual for the SCP command, and I can do manual SCP, I can do manual zcat, and this is what I meant. These three commands there, gzip, gunzip, and zcat, they're actually the same thing. That's why there is a single manual for all of them. Zcat is maybe the easiest to use. And actually on the cluster for some time there are multiple manual pages that it is able to access for the same command. So you have to press Q multiple times to exit. But it works with any commands. So the Maven, so SCP, there's also just a manual for CP. And someone before was trying to use the little r option. And I wasn't sure about the capital R, little r, but yeah, the two options do exactly the same thing here. Or you can use the full option name, which is recursive. Yeah, so the man command for manual. Oh, these are the samples. So if you do LSL, are you in the directory now? What if you first text it? Okay, LSL. Oh, sorry, LS. Oh, no, sorry. Control C. Yeah, it's asking for a log file name Q now. You're in less still. So you have to quit less first. Oh, it's actually just Q to quit less. And then LSL here. LS space dash L dash over hyphen. Yeah, enter. Okay. Oh, and just FR, just bring back the command. Yeah. Yeah, like this. And then add H. H stands for U. Okay, enter. It's going to try to make things more easier to read for you. So instead of having these crazy numbers here, now it's telling you that there are about 2 gigs. Instead of printing this 2 gigs in actual precise count of number of bytes the files are, then it's printing how big the files are. So the P10, KO, and everything is the samples.
So, I mean, the how many reads are, I mean, the first questions, the second, how many reads, it means like, how many reads in each sample? On this rate.
In the first sample.
In the first sample.
Yes. I think the question is how many reads are there for the sample P10K or red one? That's one simple name. Okay, so let's just, I'll just show you the, so the third question I'm going to leave, so the third question was, are the base call good for the second read of the first sample? I mean, this sample P10K or red one. And then, so this is the sample we were working with. So for the first question, we can do zcat, this, and then we can do head, right? And this gives us just the top of the file, right? We're just printing the files. And then the command you will want to use for questions like how many reads or how many lines is the word count command. If I do man word count, it can just yeah, it just prints a number of statistics, and so you can do word count lines. So if I go back to this command and instead of head, I use WC lines. It's just counting the lines in this file. And in principle, because this file contains a number of reads, and each read has 4 lines. This should be a multiple of 4 because the file should not have half lines. And it's going to take a little bit of time. And I should totally try to get. Yeah, so it's just unzipping the entire 2 gigs of data, but eventually it's going to print the number of lines in this file. And then from that number of lines, I can just take that number, divide it by 4, and that's going to be how many reads there are in the file, right? Okay, does this make sense? So basically, you have to kind of know these small commands that do very basic things. And the more we practice, the more you're going to discover this command, the more you're going to remember them. Yeah, so that's it. That's my number. So one thing there are multiple ways you can do to divide a number by 4, of course. But here I just loaded the R module, and then I started the R session, and we can just enter this number and divide it by 4, and it gives me this result. So that's 45 million. Okay, that's one way to just know how many reads are in this file. One trick, kind of advanced trick, I can show you for FastQ files. So this is my head, and I can just head into less, right? So these are the first 2 reads. So the head, maybe head 8. Yes, these are the first 2 reads. And one thing that's always complicated to do is because just shell is really made to work on things line by line. And then in this case, we actually have blocks of 4 lines. There is actually kind of a trick command to make your FastQs be on the line per line. And it is just something you have to know by heart. It's like paste and then 4 hyphens. And this makes your FastQ files. This just regroups every 4 lines on 1 line, like this. So this is the ID field. So the first line of the first read becomes the first field of the first line, and then the second line of the first read becomes the second field of the first line. So you have the, and then the plus, and then at the end, the sequences. So I have 2 reads, 2 lines. And then I can extract the sequences, for instance, with a cut command that lets me select columns. I can just select the 2 sequences. Right. So just one tip with a paste 4 hyphens. You don't have to know this, but it's often useful. Okay, and how long is the first read? Well, you can just, this is a sequence of the first read, so any way to count the number of characters in this line is working. One thing that is not exactly working is echo like this. So counting the number of characters in this, but the last character is actually, if we look at the actual characters, this is just something I have for myself. This is not a real command, but it's going to print what there is on the line. So this is a sequence, right? And then this is the last character. It's a new line. So when you do echo just adds a new line at the end of what it prints. And so actually, if you use echo, you have to know that there's a new line. There's going to be a new line at the end. And so it's not 77, it's 76. Yeah. The other way is to just use, if you want to do that, there's another command that you just go printf, which is going to not add that new line, and it's going to print 76. Okay, and those are the third question. I just leave it for you to work on. Yep. Okay. So yeah, this is it for FastQ file. Just go back to the slides if you need. Play with the files. The next thing I want to talk about is actually the data we're using here. So this sample names, what do they mean? And so it's from this 2016 paper. And actually it's a paper looking in mice at the differentiation, at the regulation of the differentiation of retinal pigments, so eye pigments, in either cones, which are the light sensitive, the color, the more, I mean, I guess they're less light sensitive, but higher resolution, or the rods, which are basically the cells that are responsible for night vision. And like the paper is about how mammals have a very good night vision compared to many other animals. And so they're looking at this regulatory network controlling this differentiation, and especially the role of this transcription factor NRL, which is basically provoking the differentiation into rods instead of what would be the standard path, the ordinal ancestral path for this cell lineage, which would be into codes. Yeah, so basically they look at this regulatory network that is associated with this transcription factor, and then they measure expression with RNA-seq. They have this library that's stranded total RNA, and they, in the paper, and it's a fairly big paper, they have a bunch of other data types. They have microarray. They use external chipstick data from a different study in the same system. And then they do a number of other analyses that we're not going to cover in this workshop, like de novo transcript assembly, validation by different ways. But basically what I wanted to say is that this is the general setup of the study. I'm not sure how clearly we can see on this, but these are, so this is basically a mouse embryo. I mean, just represented in time. So this is time passing with different things happening. And so it starts with like different stages like E12, E16, E18. And then here, this one here is P2. And then there is P4, P6, P10, P12, P14, 21, 28. And Yeah, and here are different, it's just different stages of the development. And at some point, the rods are, the two cell lineages for rods and cones are splitting, and they have RNA-seq for different stages. And so when you see this P10 or P2 that in the sample name, that refers to which stage the samples have been sequenced at, sampled at, and then rep, which is just replicates. For each stage, they have 2 or 3 replicates. And then they have KO, so they have this mice where this transcription factor NRL has been knocked out. And in these mice, they're not developing rods at all. All the receptors differentiate into cones, just because they can't activate this differentiation pathway. So yeah, that's basically what they're looking at in this study. Just what is a differentiation regulatory network for rods in mammals, or at least in mice. And these are just 2 small things they have, 2 small figures they created from those RNA-seq data. And so this is a PCA, and I think we're going to do one next week. It's not going to be the same because we're not going to use the same software, exactly the same methods, but it should look something like this. And you can, so this is P2, this is the P2 stages, and then one of the, so the green ones are in the bottom. The line is actually, this is the early stages, and this is the late stages, and this is for the wild types, so the one differentiating into rods, and these are the transcriptomes for the cells that differentiate into codes. So from the knockouts that can't differentiate into codes, into rods. And this is a number of genes that they have found are differentially expressed between either the wild type and the knockout mice, or between the early stages and the late stages, or only in one of the 2 genetic backgrounds, with the color representing how expressed the genes are. So we're going to do some heat maps like that as well. Okay, so this is just to give you like a bit of just what this data is and what the biology behind it was. And I think I've put the paper in the Dropbox folder as well, if you want to look at it. Oh, one thing I wanted to mention very quickly is, so this data, it was made available as with the publication, like it's like open data policy, right? And generally so if you have data and you publish your data, you're going to have to make it available. And typically what that's going to mean is you're going to put it in the NCBI sequence read archive. So you're going to have to just go to this website and then create a bunch of IDs for the different things you're doing. So this is the bio project for this study, for this data. And then it's got an ID, and then it's got an associated publication. And yeah, it's just got it's just an ID they've created in on the NCBI for their study. And then it's going to be linked with different things. And one thing it's going to be linked with is the sequencing runs themselves. So it's going to be linked with the data itself. So I think if I click on this link here, which is the experiments, and the experiments really mean the sequencing experiments. If I click here, then I end up on this page here, where I see that this is still the bio project ID, which we're on the page of just before. And these are all the experiments. So these are the samples, and there's going to be 30 of them. And I can download some table that just tells me what the samples are and like the different IDs for everything, and just like this big table of identifiers and what they correspond to. Oh yeah, and this is what I meant actually. And then this is basically so the main ID for the FastQ file of one sample is going to be an ID starting with SRR. And the SRA just gives you a toolkit, just a series of programs, one of which is FastQ dump. And this is how we retrieve the data from the study, just in case but generally, the point is, if you have to publish your data, you're going to have to put it in the sequence read archive, most likely. And basically, you're going to have to create a project ID, sample IDs, and then list all your experiments, how they were conducted. It's a more digestive methods, basically, in this sort of things. Yeah, and then provide basically the idea of the bio project is what you have to provide in the data accessibility section of your paper. How much do we have? 15 minutes. 10 minutes. Oh, 5 minutes. Okay, so I guess we're not going to just actually go through the terminal work, like actually doing this ourselves now, but I can present basically the first step you want to do once you get the raw reads is just not transform them into anything, just look at the files and at what it contains. And one tool you can use for that is this program called FastQC. And so we're going to just run the program. It's very simple to run. You're going to run it for every sample. And then it's going to give you a number of, for every library you've prepared. And it's going to give you a number of quality statistics. So the first quality statistic it gives you is just how good your bases are. So with this quality scores we've talked about, between 0 and 40, how good your bases are over the lengths of reads. So in this case, these reads were 75 bases long. And then, so the bottom half is under 20, so over 1% error. And then this is under, I'm not sure exactly. I'm not sure why they draw the bar at 28 and not 30, but this is like average quality, like good quality, where the error is between 1% and 0.1%. And this is error under 0.1%. And this is a fairly good run. Like you see that most reads, so these are bar plots of all reads. And so this is a median. The red line is a median quality at that position in the read. And then there's a bar plot around it. The quality decreases a bit over 75 bases. This is typical with any sort of sequence, at least with Illumina sequencing, actually. It's not necessarily the same in long read technologies. Yeah, and this is a fairly good run. I mean, even at the end of the reads, you can see that you're still in the green part, which is very high quality. And a bad run would be something that just like collapses after some point. And nowadays, like Illumina runs are more rare. But it's so good to have a look at this plot, check that things are as you expect. The second statistic it gives you is the average quality within each read. So this is a histogram of the mean read qualities. So on the Y is a number of reads. And then, so the average mean quality is 40 for the mode. And then number of reads have like smaller qualities, but like we don't have any reads that are really bad. Like almost all reads have an average quality above 30, which is very good, like 0.1% error or 99.9% precision, at least predicted precision. And yeah, oh, one thing I didn't mention is these FastQ scores. That's basically the Illumina software that processes the Illumina camera images that just calculates based on how good, how clear the color spots were. It just predicts that the score is this or that. And then this would, on the bottom left, this would be an example of a run that would have a number of reads that are weird, just because they've got this like number of reads that are clearly not as good. And same thing, I think, on recent Illumina sequencer, doesn't happen that much anymore. This is more important. So this is just the percentage of each base that is seen at different positions in the read. And here on the left is actually a good one. And it's kind of tells us that this is like it's got this exclamation mark, meaning it's not passing all the filters that FastQC internally applies. But it's actually normal. Like it's because of this like weird patterns at the beginning. But this is actually because of the biases in where your primers are going to bind in the RNAs. It's not exactly random. It's not fully random. So you'll very often have some bias at the beginning of reads for RNA-seq. So this sort of thing is not unexpected for RNA-seq, and then it becomes flat. And this is a recent sample I had to process, and which eventually, even though I kept the sample, FastQC in general just gives you an idea that some sample might be bad, but unless they're really, really bad, it's still maybe worth it to just go ahead. But if then later in the pipeline you have other indication that they might be outliers and have technical issues, then you know that even at the sequence level something was wrong, and you can just remove them and flag them outliers with good confidence. And yeah, this was just weird, and it really was supposed to be like this, and this is what it looked like, and with this weird biases even later on in the read, which didn't look like the other samples of the same batch. And then it also gives you the percent GC per read with this blue curve, which is the expectation, and then the red curve is what is observed in the sample. And in principle, they should be kind of overlapping quite a bit. And this is this bad sample that I had, which was really weird. And this is like something that just tells you that your sample pre is not usable. And then the last thing, I think there's one more. No, the last thing is this overrepresentation analysis. So it's going to look into the data set and then tells you if there are things that are highly overrepresented. And typically in RNA-seq, because like, I don't know, housekeeping genes, for instance, they're going to be, they have very, very high expression level. So it's pretty much always going to tell you there's a little bit of overrepresented sequences, but it shouldn't be something large. I mean, the best thing is usually to just compare the different samples in your batch. And if there was an issue with library preparation for some of the samples, then they're going to look different. And the numbers are not going to be the same. And also in this case, so they have this very highly overrepresented sequences, like almost 10% of the data is actually this sequence there. Or this sequence appears in 10% of reads. It's actually the Illumina adapters. In this case, would be adapter fragments. So instead of sequencing an actual fragment of reverse transcribed RNA made into DNA and then ligated to adapters on both sides, the adapters have just been ligated to one another, and you're just sequencing the other adapter instead of sequencing something meaningful. Okay, so this is we're going to so next thing we're going to do is just go through this FastQC analysis. It's easy to install, so you're going to download it, and then we're going to run it on our first sample, and that's going to produce this. Actually, these plots are produced in an HTML document, so very easy. Yeah, so that's the next thing, quality check for tomorrow. Okay, do you have any questions?
So we're going to do FastQC tomorrow?
Yes. And yeah, try looking at the FastQ files. If you're not very comfortable with Unix command, just practice a bit. Try answering this question on the qualities of the second read. Are they good or not? So just go to the quality line for the second read, and then go back to FastQ Fred score slides and figure out whether they're very good or not. Yeah. Okay. Well, that's it for today, I guess. Yes. Oh, you had a question, or no? Sure. Which one?
No, it's just that the copy never actually.
Oh, it didn't end. we'll have a way to deal with that. It's not going to go away. And if it didn't work, I can show you ways that you don't actually have to copy things in Unix. You can just make false copies that let things be in your directories, but also in another directory. Anyway. This was the time we had, right? Yeah. No, I mean, I'm staying here, and I think I have to, thanks to the online participants as well. I think they were happy. And then I should just stop the recording. I think I have stopped the, okay, stop.